Everyone, it's 11 and we're going to be running into lunchtime, which I hate. So let's try to pick it up so that we can all have lunch early. So you are a diverse crowd of people, like in general, but also in terms of your the things that you've studied before. Some people are coming from computer science, others are coming from psychology, others are coming from biomedical sciences. And so what this means for the course is that we're trying to get you all up to speed in terms of, that you all have a sort of the same level of going into the course, right? So that's what we're taking these couple of weeks for. That's why the first practical is mostly just Python rehash, right? And how to open an fMRI file and how to conduct very simple analyses. So if you have a background where you haven't emphasized programming in Python and doing data analysis sort of things, then make sure that you start spending some time on that practical already. Yeah, you can ask questions on Discord. The TAs will be monitoring that also. So if you have any specific questions about specific segments of that first notebook, let us know. At the same time, you might have like a very computational background and you're like, okay, all of this psychology, biology stuff, I have no idea. And for that, we have this lecture. And so you still have to go through that first practical, and I recommend that you do it early. But, this lecture is just to bring you up to speed in terms of general neuroscience background knowledge, right? So what I mean by that is, today we'll be running through more or less like the things that you need to know in order to not be completely, is the word stymied or stymied? I don't know. Anyone? Any native speakers? Because, you just read these words and you don't know how to pronounce them because that's English, right? No? Stymied? Yeah? Because I've said Ari, the word awry, which you shouldn't pronounce as Ari, but I have. So yeah, that's one of those words. I think it's the Y, it can be anything, right? Sorry for the digression. So today we'll be going through a modicum of neuroanatomy, neuroscience background. And the goal is really to give you an idea of what the hell is in this figure, right? So I have a rotating brain there. That's all nice and good, but what is in it, right? So, for example, just to give me an idea of where you guys are. What's green here? What do you think is green? What's colored green in that image that you see rotating there? You can just say anything, right? If you say leprechauns, that's fine too. It'd be wrong, but Yes, that is a very good point. So what you're seeing is green here is the central sulcus. On the anterior bank of the central sulcus is the motor cortex, and in the posterior bank is the somatosensory sulcus. So specifically what I've made green here is just at the sort of backside of the central sulcus, which means this is somatosensory cortex, but excellent suggestion. So what's red here? Anybody know? Exactly. So what we're looking at here, this lobe is called the temporal lobe because it's up near the temple of the head, right? It sort of arcs down like so. It's where different things are. It's very memory related and things like face selectivity. When you see a face, the regions in the temporal lobe activating, but specifically, like you said very well, this is the superior, right? The upper part of the temporal lobe. And that's where a specific sensory modality lives. Anyone have an idea? Sorry? Excellent. So this is auditory cortex, right? So when you hear, the cochlea invents the sound for you, and it ends up and is colored red here. And then, of course, maybe blue is easiest here. Any idea? Exactly. So that's the occipital lobe at the back of the head. It's where another sensory modality lives. Exactly. Yeah, so that's where vision lives in the brain. And so what you're looking at here is the result of a specific analysis that is meant to show how these things intermingle in the brain. That's also why you see certain patches be red in what we call the frontal lobe, right? There's a bit of red there. These are language-related regions that have to do with your comprehension of speech, for example. Yeah. I'm just going to muse a bit. So this is mostly very unstructured lecture. I'm going to go from small to large during the lecture, but I think it's just nice as if I just muse and we have a little conversation about where you guys are in terms of background, and you all get to be sort of in the same place. So I have delineated things on the surface here with these lines. Anybody know what we call where these lines lie in the depths of these folds? Yeah, so what you have is this folding pattern, right? When you fold something, when you crumple like a blanket or something, certain parts have to come up and other parts will go down, right? The parts that go up are called gyri or gyrification. A gyrus is just a fold, one that moves up. And if things move down, so if you're at the bottom of a fold, we call that a sulcus. So what we have here is lines running through the major sulci, because it's a Latin word and we try to be very expensive with our wording, of course. The plural is sulci, and that's why we put them there, right? So it's a nice sort of delineation. The idea is that with a lot of analysis techniques that we perform, we can actually blow up this sort of surface of the brain. And if we draw a line somewhere, then we still know what was at the depth of the sulcus, right? We don't lose that information by just delineating things like this. Yeah. And so what we're going to do now today is just go from the smaller parts of the neural architectures of our brain to the larger parts, and end up somewhere here, right? So somewhere where you have an idea of what the different lobes do, what their major functions are, and so forth and so forth. Cool. So the major trajectory here is going from small to large. One thing is, in my mind, maybe I'm a bit biased. I was once trained as a biologist and physicist, so I tend to find technical stuff really interesting, and I find living things very interesting. And so my focus is generally also on how the brain is like a living machine, right? It's an organ. Yeah. And what that means is you've already seen that in the first lecture, of course. You see, for example, things like pulsatility, right? You see the brainstem pulsating, right? It's a beautiful example of how the brain is just a living organ that we're trying to work with, that we're trying to take measurements from. Yeah? And so if we zoom into that sort of biological aspect of things, I'm going to first highlight a couple of things. So different components of these microscopic brain parts. The first being neurons, then we'll go into what we call glial cells, and then we'll go into vasculature, all still at the very microscopic level, because these types of constituent parts of the nervous system will mean that we pick up different things with our MRI scanner. And knowing these things is very important to be able to interpret what we're seeing in a scan. So you all know neurons, right? I mean, more than half of you is AI, so you know what a neuron is. It's this thing that has an input side and an output side, right? And it performs a very simple computation that we know also is an aspect of artificial neural networks in many cases. So you have an input side and an output side, you do some computation in the middle, and then you send out a signal. So the connections between these neurons are called synapses. Many of you will think this is a bit boring, right? But I just want to go through all of the different steps. So synapses are these contact points, and they are very dynamic biological contact points, right? There's just proteins that make up the synapse that make sure this connection is, for example, stable, and that you can grow new ones, that you can learn specific things. Yeah? So what happens is the signals that the neuron takes up from the dendrite side, that's the input side of things, it pools the currents that are input there in this dendritic tree on the input side, and then as a certain threshold is reached, it projects a signal outward. So what that means is that there is a wave of current that gets transmitted along the axon, and then when it reaches the output synapses, it transmits that signal to the next. Now, I'm not going to go into the action potentials themselves necessarily, because that's a bit too low level. I just want to go into like the cellular composition of what we have in the brain. And this is an important one because we think that these action potentials that the neurons send are really the thing that makes up neural function, right? So when you experience something, we don't think that it's the vessels, the blood vessels that encode this experience. We think that it's the firing of the neurons that is the preeminent sort of biological substrate of what we experience. Yeah? That's really the sort of dogma. This may not be entirely true, and we'll get to certain parts of that in the next lecture also, but this is really the dogma. When a neuron fires, that is a sort of ingredient, like a little Lego block of what you experience, what you see, what you feel, and so forth and so forth. Yeah. And what that means is that these, for example, these glia cells in the vasculature are not thought to take part in that. Now, what I'm showing you here is just an example, again, of how that neuron is part of like a biological organ, like a matrix of different things. So what you're seeing here on the left is neurons firing. So this is a little patch of hippocampal tissue in the hippocampus. I'll go into what the hippocampus is later. And these cells have been infected with a virus so that they express a protein that when the calcium concentration changes in the neuron, you can see it. So when one of the salts in the neuron changes concentration, you start to be able to see it with a microscope. And so what you see here is that different neurons in that hippocampus are firing. They're firing these action potentials. They're taking in signals and they're projecting them out. I will separate out these movies and enlarge them a bit for you and then upload them to the file so that you can have a look. This is really magical. You're really seeing these cells fire, and people are developing more and more elaborate techniques to be able to do this in animal models. So this is really a huge frontier of neuroscience, is to be able to perform recordings like this. So that's this calcium imaging that you're looking at there. Just one of those imaging modalities that is also at that spatiotemporal graph that I showed you of different measurement modalities. What you're seeing here is a microscopic movie of how neurons intermingle if they are put in a petri dish. So you see that these neurons are like little critters, right? They just sort of crawl around and they try to connect with one another, and they're very dynamic in this sense. And this is also something that you need to sort of keep in the back of your mind as you're thinking about just imaging the brain as a living human brain. It's composed of all of this sort of thing. Yeah, it's composed of these cells that are pulsating, they're living, they're trying to communicate with one another. They are dynamic in a very, very fundamental way. So you're not just looking at a voxel in an image. What you're looking at is populations of these neurons firing, cohabitating with one another, trying to transmit information with one another, and it is a zoo out there, in there. Just so you know, right? Now, neurons may be considered very important for information processing, right? They are the sort of dogmatic element of information processing in the brain. But they are definitely not the bulk of the tissue in the brain. The bulk of the tissue in the brain is actually these glial cells and vasculature and things like what we'll go into later, the cerebrospinal fluid. Yeah, so these may be sort of the more important ones, we might think. These need all the other stuff to remain alive, even. So what are these glial cells? Well, they're sort of the roads and the plumbing and the sewage of the neurons, right? They exist to form a matrix around the neurons to nourish the neurons and to keep the neurons alive. It also means that, for example, when we see a bulb response and we think that we're looking at a neural response, we're wrong, because the response that we actually measure is partly mediated by these glial cells. Yeah. So even though we might not really think that they're very important for information processing, we think that the neurons do that, they are an important constituent of our measurement, and we need to be aware of this indirectness of our measurement yet again. Yeah? So you have different types of glial cells. You have large ones, small ones that make up the sort of blood vessel, sort of size of the blood vessels, for example. You have these ones that are just sitting next to neurons and giving them signaling molecules and trying to feed them. And we have very specific ones, which are these Schwann cells that I'll get into in a bit, because we can actually pick them up very nicely with the MRI scanner. Yeah. Now, this is as micro as I get. I won't go any further into the sort of biological detail of cells much. So if you have any questions about that specific type of thing, let me know. You can also ask on Discord and whatever, but I'm just going to go to the next slide. So here's your opportunity to ask a question. Great, because we're here for fMRI. We're not here for cells. Now, this other very important component of what's in your head, that's not neurons, is this thing vasculature, right? And as I've already said, when we do fMRI, in most cases, what we are looking at is the blood vessels. We have just zoomed into what those blood vessels look like. You have a supply side and a removal side. Yeah, the same as, I don't know, the liver would also have. Yeah, the blood needs to come in from the heart and the lungs, providing fresh oxygen, and then it needs to take all of the crap away. Yeah, but in the brain, it's quite complicated. The brain actually, of course, uses up most of your body's energy demands, right? So the vasculature is really important to provide all of that energy to all of the neurons, both in terms of glucose and in terms of oxygen. And we'll be focusing most on oxygen. So what does this look like? Well, you have your arteries on the supply side and your veins on the, removal side. And what's in the middle between the larger arteries and the larger veins is the neurons. Yeah, so your gray matter is here. You can see that here on the left also. So this is the gray matter of the brain where all of the neurons live. This is the white matter. We'll get into that in a bit. And you see that there's large, very large veins on the surface of the brain. We're talking here about the cerebral cortex, the outside of the brain. You see these large vessels, both arteries and veins on the surface, and then they go down and project downward. And in general, what you can see is that along the gray matter, there is this capillary bed. And all of these different sizes of vessels that you see here, so going from the large to the small back to the large, all of them are dynamic. So again, this is a living biological type of tissue that you're looking at. The arteries, as you see here, they actually have some smooth muscle tissue in them. And what this means is that it's not just your heart that's beating, the arteries actually help to sort of draw the blood along. Yeah. So this is really trying to push blood into those capillaries to provide the neurons there with their oxygen and glucose. Yeah. At the same time, what of course can you do on the removal side? You can, instead of push, you can. Exactly. Okay, and the exam questions will be harder than that. So what can the vasculature do to pull? Well, you can, when you have a pump, right? You can widen one part, and that will decrease the pressure there, and then suck things into that direction. And that's what happens with the veins. They dilate as you have neurons activating, right? So you have some push and you have some pull. And as you go, this is a standard thing, right? So when blood is oxygen rich, it's very sort of cherry red. And when the oxygen is depleted, it changes color and it becomes more bluish. It's just why people that suffocate get blue in the face. And that's what we use here to delineate this gradient. Yeah. These capillaries are really, really, really tiny. The smaller they are, the better they can exchange this oxygen with the surrounding tissue. Yeah. And so this Bold response that we measure, that I've spoken a bit about and that the next lecture will also be about, is really this beautifully complex cascade of things that happens in this vasculature in response to neural activation. Like a wave of things that are happening in terms of pushing, in terms of pulling, and in terms of the effects that this dynamic has on the amount of oxygen in the blood. So it's a very complex biological sort of machinery that we're looking at there. Beautiful. And when we summarize it in a single voxel, of course, we're losing a lot of information on what exactly is happening. We're summarizing that with that bold response that you've already seen and that we'll talk about more. But it's important to realize, right, all that we're looking at with fMRI, you do it in a standard way that you'll all do if you ever do it, is this bold response, a blood oxygenation level dependent response. It's all blood. right? And so you're looking at this blood organ thing when you're looking at fMRI. So, yeah, we tend to think like, this is cognitive faculty here in the brain, but you always need to realize that when you perform an analysis, the things that this came from is always this blood response. It's important to keep that in the back of your mind. Now, in 1 week, I think on the 22nd, is that 1 week? I always forget these sorts of things. Maybe a couple of weeks. You're going to get this MR physics lecture, so you're going to get a better idea of how these magnetic properties of tissue give you specific image characteristics. I showed you these two different types of images, a T1 weighted and a T2 weighted scan. You'll learn a bit about how that works. Now, in a standard T1 weighted scan, which is our standard anatomical scan, a brain would look a bit like this. And it's easy to interpret this T1 weighted scan. Again, don't worry about the word there, because the brain looks more or less the same in one of these scans, these anatomical scans, as it would look if you would take like a sword and just cut someone in half like so, like in Kill Bill. So what you have is you have white matter in the middle of the brain here, and you have this gray matter in the outside of the brain. Same, it looks the same in one of these MRI. So the white matter is composed of the axons, so that is the bundles of axons going from one region to the next. So these are like highways between brain regions. And why are they white? Well, they're very fatty. Why are they very fatty? Well, they are, this white matter is composed of these Schwann cells that you already saw on one of the previous slides. They really wrap around the axons. They're very, very fatty substance. They have a very, very fatty substance called myelin, which sort of insulates electrically the axon. Now, what this means is that the signals that propagate through the axon, they don't have to go millimeter by millimeter or nanometer by nanometer, but they can hop over. They can just start here and then immediately hop over there, there, there. And that accelerates the transmission of the action potentials tremendously, but like enormously. So what does the brain do? When it notices that a certain bundle is important for survival, or just important, what it does is it starts to invest in setting up these myelin sheaths for a specific axon. And we see this also in things like brain maturation, right? We see that myelinization increases in specific brain regions in specific periods of development. It's because what the brain is then doing is it's trying to cement the function of these specific axons by myelinizing them. I don't know if that's a word, but you get the point. It is like accelerant for the potential transmission, for the information transmission. And so the brain invests in it to make certain types of communication faster than others. Again, a biological process that takes part of learning, if you will. At the same time, you see this gray matter on the outside, right? That's that curved surface, the sort of thing that you're crumpling up inside the skull. And that is what contains all the cell bodies. So the neuron bodies, they're somas, as they're called. And so this is where all the energy of the neurons is being used. It's also where, if you do standard fMRI, you will see activations. If you start seeing activations in white matter, something is wrong, usually. We can go into specifics how sometimes that's not the case, but in standard setting, if you see signals appearing in white matter, something is wrong with your scanning, with your analysis. So in fMRI, the white matter is silent, and the gray matter will give you where the blood is, that's where the neurons are, that's what will give you the signals. Clear? Excellent. So this is all nice. I've only shown you cartoons, right? But again, I want to highlight this sort of organic nature of what we're doing with the brain sciences that we're trying to perform on living humans. So what I'm showing you here is my head, but sampled with very, very tiny voxels, because as you decrease the voxel size, as you increase the resolution, you're starting to see this biological detail much more, right? If you just have a very coarse scan, your brain, the brain that you see will just look like more or less a standard brain. You will not see the idiosyncrasies. You just see gray matter as a single gray matter sort of line in the brain. And you won't be able to, for example, make up a lot of anatomical detail because the voxels are just too small. And what I'm showing you here is just an example of what you can see just in a living human brain, right? Trying to take a step up. We're not looking at cells anymore. We're looking at like very, very tiny tissue properties. And I'll show you a bit of what happens when you start zooming through this. You start seeing a level of detail that you don't know from like the standard anatomies. But it's important to realize that all of this detail is in the head, right? So you're already sampling it, even though you're not seeing it, if you're blind to it with larger voxels. Yeah. So what I'm going to do is just click, and then we'll start sort of zooming through these two scans, or at least one at a time. And I'm going to try to highlight things like veins that you see very clearly running through the brain, and things like the layers of the cortex, right? Because it's not just one band of gray matter. We actually have different layers inside the gray matter that we might be interested in also. So this is slowly going. This is actually the back of the head, and it's, I think, a coronal. You all know now, right? I think it is a section. So this is towards the back of the head. And I'm actually combining a couple of different types of scans here. Let's stop. It's supposed to loop. Let's do it again. And so what you see here is that in white matter, you see all these vessels. You see these sort of Star Trek-y things. All of those are vessels. So I'm trying to show those. They become purple in the scan that I'm using here. You see a lot of that vasculature just running through the. Do it again. You're going to see this is a vein, and I'm tracking it. You see that it sort of snuck away like that. And you have another one here, another one there. You see these vessels really popping up like between two layers of gray matter, for example. Yeah. Now these are these vessels that will give you enormous signal, but they don't really mean anything specific, for example. What you also see here at the bottom is the cerebellum. I'll talk about the cerebellum a bit later. You see that this is the cerebral cortex, the upper part of the brain. You see it has nice detail. For example, you can make out lines here that are these layers of the cerebral cortex. You see then that this is much thicker. This is about 2 to 3 millimeters. This is much thicker than the detail in the cerebellum, which means that we are completely blind to relevant signals in the cerebellum if we do standard fMRI. You're always going to pool a lot of these beautiful sort of tree-like structures together if you're going to sample standard fMRI. So what does that mean when you say something is activating here? Do you even know what that something is, right? It's important to always take a step back and try to realize, what are we measuring? Yeah. So let's go again. You see beautiful sort of arborizations here. You see the vessels there. You see these huge vessels really on the side of the brain. You see a lot of structure there, right, that you might not see if you look at it with less detail, but it's always there. Now, similarly, here's another one going from the bottom to the top. I think this is my neck. Like, this is muscle. You see there's veins in muscle too. And at some point you'll start seeing the cerebellum. Here you have it. That's just another view. It's beautifully tiny arborizations, right? White matter and gray matter in there. And here you see the cerebral cortex with its white matter and gray matter. You actually see lines in the gray matter here, so a little whiter stuff inside the gray matter. You actually have different layers in the gray matter that have different levels of myelin, which is why certain parts look lighter than others. Yeah. So there's a lot of beautiful structure, even in a living brain, if you care to spend the time in the scanner, like for an hour with a bite bar, not swallowing for an hour. But then you can get this sort of thing. Yeah. Here I'm combining different scans together so that you get an idea of these veins and stuff. But yeah, you get the point. Yeah. I've uploaded these movies too, so you can sort of scroll through them and see. You sort of like trace the vessels, right? And you're always going to have those. Now, quickly, let me just run through a bunch of, really basic. Yeah, go ahead. Sorry. Yeah. No, please. Always ask question. Anyone. Yeah.
About the video. Like, what were the light blue parts? Like the purple ones were the veins, but the blue ones were the.
Yes. So what I'm doing in this scan, which is a bit difficult is that I'm showing you one T1 weighted scan, which is why the white matter looks light and the gray matter looks dark. And what I've done is I've superimposed another scan where it's more sensitive to blood vessels, so that I can sort of make them pop out specifically. That's called a T2 star weighted scan. You'll get a handle on what all these things mean, but that's also what we use for functional MRI. So if you use it in an anatomical MRI, you have just one image, and you're very sensitive to these veins. So that's really where what you're seeing here is really where the values of that specific scan they dip down very strongly. So it's exactly the same as an fMRI, right? If you have a vein, signal goes down there. So that's what you're seeing. And you'll see that it just whenever it goes through a certain value, it becomes like greenish, bluish, and then it becomes purple if it goes further down. That's about it. It's not trying to do anything fancy. I just played around with it and I thought I saw a vein, so that's it. Yeah.
Okay.
Cool. What you also see is that specifically in cerebellum, everything is very, very crappy, and you just see a lot of noise everywhere. Look at it; it's just fucking noisy, right? It's like you're looking at an old, like 40s movie sort of thing, right? And that's really also what we always have to deal with, right? Not just the fMRI can look horrible. We try to push the anatomies, it will also look horrible. It's a strange thing to say about the inside of your own head, but still. Okay, so major portions of the brain, like in a macroscopic level, right? We've zoomed out further and further, and then we'll start going into function a bit after we have a tiny break, I guess. So we divide the brain into different lobes. Most of the brain has a left and a right hemisphere. Only certain portions in the brainstem are not bilaterally symmetric. And then when we take the cerebral cortex, for example, that's this part here. You can divide that into lobes very broadly. So you have what you already said, right? The occipital lobe, the temporal lobe, the parietal lobe, and the frontal lobe. We missed the parietal lobe, but that's the blue one there. And here you see some specific functions that we associate with these different lobes. I'll go into the specifics of these functions a bit more later on in the lecture. But we can also look further down and then think of things like cerebellum that you just saw, right? With its beautiful tiny arborizations, and then you have the brainstem. And just in general, these are more the evolutionarily older parts of the brain, so they do more what you might call primitive functionality, and we think that our truly miraculous human mental capacities all sort of live here, right here at the top. So things like the frontal lobe, it does abstract reasoning, planning, that sort of thing. Parietal lobe does spatial things like how do we navigate through space, that sort of thing. There's a lot of language also in the parietal lobe, and so forth and so forth. Yeah. So this is very old school. And a lot of the original ideas that we have about how the brain is organized come from things like lesion studies, where you notice a patient, they have this and this problem, and then you notice later on when they die and you cut them up that there's a hole there, right? And then you say, apparently this is what made that go wrong, right? That is a very, very coarse way of thinking about brain organization, because it makes you prone to really say, this function is there. Yeah? So this separation of function into the different lobes is a bit old school because of this bias for just pinpointing a specific location and saying it's this function. What we think nowadays more and more, and we'll get into that as we progress through the course, is that the brain really is a very complicated network where these subparts of these different functionalities are really intimately connected. It's becoming more and more, well, harder and harder to keep saying this specific function is there, because you can find evidence of that function all over the place, even though it didn't show so strongly in these earlier lesion studies. But let's not get ahead of ourselves. Yeah, go ahead.
Go back to the last picture.
Of course.
Why is the cerebellum, why does it look so much different than the other lobes?
Yeah, so it's evolutionarily, it's from a different sort of arc. So if you go back to animals such as sharks and like about like where mammals started, you have this thing, this thing, it's called the telencephalon in these terms. It used to just be the little part of the brain that these pre-mammals would smell with. And that sort of mushroomed into this entire cerebral cortex. Well, at that time, there were also other regions just in the brainstem and on top of it, such as the cerebellum, a very old region evolutionarily, and it just had a different function, which, for the evolutionary repertoire that made this region, the cerebral cortex, grow so large, it didn't have the same effect on the cerebellum, although we do think that cerebellum is incredibly important in varied things in terms of cognition. We'll get into that later on in the course. I keep saying that, but it's true. Trust me.
Is this only for humans then, that the cerebellum and the motor is much different compared to other mammals?
No, all mammals have this specific basic architecture. It's just that, for example, in mice and rats, they don't have this specifically strong enlargement of the cerebral cortex because, well, we can pretend that they're simple animals, right? And so relatively their cerebelli, Latin word, are a bit larger. But if you look, for example, at a dolphin or an elephant, they have like incredibly huge cerebellar cortices. And if you're interested in this sort of thing, there's actually a beautiful project by a scientist called Roberto Toro that you can look at. They have they've scanned, I think, something like 100 or 200 different animal species in an MRI scanner. And so they have like atlases where you can really go through all of these different things. They're like a browser-based thing. It's gorgeous. Yeah. Questions? Other questions? I'm just going to continue for another 5 minutes or so, and then we'll have a break. Trust me. So there's a lot of words on this slide, and I'm going to quiz you on all of them, which is ironic. But what this serves to show you is how not the major lobes of the cerebral cortex or cerebellum are involved in different mental capacities and types of cognition, but how these lower layers of the brain are. This information may not be too much fun, but let's go through it because it's kind of interesting nonetheless. So these are relatively old parts of the brain, like we said, like mammal, pre-mammal. Probably find something like a shark. I did biology, so I dissected all these different animal brains, so I should know, but it's been a while. So yeah. But so these middle parts of the brain, they're older, and you can think of this as the higher levels have been just sort of stacked on top, right? So what that means is that when you look at a specific type of function that is being performed by a specific lobe of the cerebral cortex, for example, it will mostly connect to a bunch of these lower subcortical, as we call them, levels, and very strongly interact with them. So first, I'm just going to run through a few. So what's important to know is basal ganglia. In the front of this this sort of we don't call it the midbrain, just a bit on top. They're all of these regions, quite a varied bunch. And in general, what they do is they regulate action and emotion. And so whenever you decide to go for something or not, this trigger is usually implemented in these regions. And so in diseases like or disorders like Parkinson's, for example, it's the functionality of these basal ganglia that is wrecked. In very specific regions that I won't go into, it's very, very interesting. But it's these regions that do that sort of motivation part of our daily existence. Yeah, things like if you're a heroin addict, you're probably fucked up in the basal ganglia somewhere. Yeah, I'm not saying not probably, definitely, but in very specific ways. Right, and a bit lower, we have these midbrain regions that do things that I'll get into in a couple of slides. Again, emotion regulation, things like arousal, that type of really sort of quasi-hormonal function that has to do with motivation and emotion, come from these lower regions in the midbrain. Now then we have the archicortex. So the cerebral cortex that I was talking about, that's all the way over here, right? We call it the neocortex because it's newer in evolutionary terms. And it was predated by what we call the archicortex, which stands for old cortex. And that has a similar structure to this neocortex, also in terms of layers and things like that. There's specific regions like the amygdala and the hippocampus that are in that archicortex, primarily in the medial temporal lobe. So that's the middle of that temporal lobe. And they're very much important for things like memory and navigation. So you'll see that happening a lot. You may have heard this sort of popular science finding of London taxi drivers having enlarged X. That X is the hippocampus because they know exactly what the map of London streets is like, right? So that's the type of thing that hippocampus thought to do. We think that it does much more, and we can talk about that more. So what I'm showing you here is really, again, like an old school sort of separation of responsibilities that we think exists. But it's in general much more complicated than this sort of cartoon portrays. Now, lastly, the thalamus is very important. It's this this reddish thing here. It has many different nuclei in it, so different sort of subcores, if you will, and all of those subcores have a different sort of sensitivity or a different role. So, like we said, the occipital lobe likes vision. There is a specific part of the thalamus that also likes vision. It's called the lateral geniculate nucleus. That's for vision. Then you have for sound the medial geniculate nucleus. So all of these different layers in the thalamus, they do specific things like audition, like vision. There's also other regions that do more memory-related things, more postural-related things. The general idea is that for any region in the thalamus, it projects to a region in the cortex, and it is more or less its relay station. So what that means is that, let's take the example of the lateral geniculate nucleus. I'm a vision scientist, so it's my nice example. What happens is light falls on your retina, and it passes through the lateral geniculate nucleus to reach this neocortex, the occipital lobe of the neocortex, where all the complicated functions of vision are implemented. Right, so it's a pass-through station for a specific type of information that's also used in the cortex. Yeah. Okay, there's so much information. Oh yeah, go ahead.
How do you get these annotations in the first place? Do you use fMRI, or was there some other technique that was used?
Yeah, that's a good question. So primarily, like traditionally, cutting people up. Right, and then you have things like histology. You have different types of cells that people would use to differentiate between these regions. For specifically for these sort of central brain regions, they're actually very well defined. So you can really say, oh, there's a little sort of circle here, and it does a specific thing because you've seen this like in an animal where people cut it out, or when you have a patient that has a problem there. So that sort of processing. If you have a nice anatomical scan, we have these atlases because they're such well-defined, nice regions. You don't need to like shift the atlas much, and you know exactly what was what. So there's beautiful ways in which you can automatically say, this is the pulvinar of the thalamus based on an anatomical scan. So that's what we do now. You can do fMRI in these regions. It's done. It's not as easy as in the cerebral cortex because the signals tend to deteriorate as you go down. That's not just due to the blood vessels pumping, but also because you're farther removed from the machine that picks up the signal. Does that answer your question at all?
Yeah.
Cool.
Why do the signals deteriorate when you go?
Yeah, so there's different reasons for that. Things like blood vasculature, like the blood pulsating there. So you just get a lot of noise that we don't want, right? We want to separate that from the signal. But also in terms of just the image quality, you have to, so you'll get this in the physics lecture, but you have like a whole array of like 64 or 32, these little copper coils placed around your head, and they're like radio antenna. So they pick up signals. And of course, as you know from like radio transmissions, right, as the further you get away from a source, the harder it is to pick up the signal. And so this happens also in the brain. When you try to pick up a signal from the middle of the brain, you're in trouble because, you know, it's just far removed from that coil that you're using to pick it up. You're using multiple coils, so you can sort of resolve this. You can get images there, but you lose something as compared to being very close to the coils themselves. Yeah. So the outside of the brain, you're in luck because you just get a lot of signal there. That's a general thing. Yeah. Any other questions before we have a break? I shouldn't have said before we have a break. Right now, everybody wants to break, but you had a question? Or are you just planning on leaving? Okay. Cool. Let's have a break. Let's be back at 55. Excellent. Yeah, we're putting them up week by week.
I was wondering, what sorts of all of them and
Yeah, so we're going to be possible, but within the realm of. So we're going to do a ride. We're not going to do like EEG analyses, primarily because the master's thing that we're coming from already has separate EEG. So what we're doing is we're trying to give like an overview of how you do standard analysis. For example, if you were conducting an experiment where you have like a group of people with schizophrenia, you do an experiment, how do you look at the differences between the schizophrenia patients and the controls, like a sort of clinical type of that's a standard type of analysis. You can also do things like information mapping based on the MRI, so that you can look at like the mental processes, how they transpire. And another focus that we try to have is like you can take all these different voxels that you've measured, right? And really start seeing them as a biological measurement that you're looking at, right? So that could be, and the analogy is then that you're likening it to like if you put an electrode in an animal, you also measure from a population of neurons, probably, and that's what we're doing here too. So trying to take that and
So it's also more other the results and how we.
yeah, very practically oriented, but we have like the clinical or AI side of things, and then those sort of biological side. That's sort of.
We have a mini.
It's like the week thing.
Yeah, so I've been back. And I was wondering
Cool, yeah, yeah, send me an email and let me know what exactly that is, and I'll start thinking about how, because this would have to be like very practical, right? So, yeah, I'll start thinking about what
really like the lecture so far. Presenter compared to other. Yeah. Now, but I was wondering, so on Tuesday. Wednesday. Okay. Either way, I have a different lecture that overlaps. So I was wondering, do you upload this lecture somewhere? Okay.
you're like the third person to ask. I have been very hesitant to send out the recordings because I really like
Yeah,
like the lecture.
like, so I noticed that in the first period, there was also some science and she screened everything and it was totally empty. And I was also not a big fan of that. I was complaining to her afterwards about that.
Yeah.
So I can totally see where it's coming from. So yeah, I was just wondering, like, if you even record it or stream it or whatever.
So what I can, so you definitely have the PDFs, right? So you can read
Yeah.
through the PDFs separately. I have all of my lectures from last year. And more, they're on a YouTube channel. If you send me a message on Discord, I can send you a link to the, so just playlist. I go through all of them. So for example, I have a video of this specific lecture. So you'd be able to more data.
Okay.
You would, I can't promise them that that will be exactly the same for all, lectures. Well, that's like my cop out sort of, answer. If you can gather a bunch of people that you find it really important that I do this for this specific reason, I will try to see what I can do. But like it's a big time investment to set up the recordings.
Yeah, no, and I think it's also nice for most people to come here for you speaking from the full crowd. So I don't want to not also, but like motivate you to do that because I really dislike that. It's instant nice people to stay. but I'm just like having walking up the course.
Yeah, I know.
That was.
Just send me a message on this and I'll think some more, because I've heard it from others. They have up until now been, very happy with it. So YouTube maybe.
The last year's video mixing, are you open to sharing with everyone?
Yeah, I mean, there's a back, yeah, I mean, yeah, they're open, so you can find them and stuff, but, yeah, I am a bit afraid that I'll be talking for like 10 people when there's 150 people in the campus. That's something that I want to make sure that. Yeah, so there is a difference between what I'm thinking now.
No, that was fine. But, thank you very much.
Yeah, just send me a message, I need to tell you. Also.
Actively be uploading. And then we start. So, but
Yeah, for example, in the middle parts of the brain that I'm highlighting here, there are neurons there. Yeah, although the white matter itself aren't.
white matter is just a solid fatty,
Yeah, it's like bundles. Yeah, like bundles of atoms.
so no neuron.
You can, yeah. So there was a reason years ago where they used different measurement techniques, like using MRI for different methods, where they found that this little white matter tissue that also shows some form of activation. But it's really not clear what that's based on.
So if I understand correctly, most of the activity happens on the surface of the brain, where the brain is, and probably some in the middle, where there is no.
But whenever you have this water, like cerebral spinal fluid, we call it, or whenever you have white matter, you don't really expect any. I'm running for a coffee, so I'll be maybe 1 or 2 minutes late for my own deadline. It's fine.
Yeah. I don't know. I feel like it's.
Also. Because I introduced myself like.
So, a lot of people are interested in specific lobes, and the lobe that most, I've looked this up, I've gone through the literature and started looking for, okay, what lobe is researched most? Ironically, it's also the lobe that we still know least about, but it's the frontal lobe. So I thought it'd be a good idea to just introduce you to what scientists at present think about the architecture and the sort of gross divisions inside the frontal lobe. Because as you read a bunch of literature, you will get a lot of these references where, you know, fMRI finding something in the frontal lobe. And it's huge, right? Look at it. And it's huge in humans relative to, for example, non-human primates, because, you know, we have this evolutionary niche where we need to plan a lot. It's more or less it. Yeah? And why does the frontal cortex do planning? Well, you can think of this from sort of evolutionary thing, like this whole bulge it grew recently. What was always there is this motor cortex. Right? We already identified the central sulcus, right? It's this sort of dividing line here in the middle, and it separates the back of the brain, parietal lobe, from the frontal part of the brain. It's where the motor cortex is in the frontal side, and it's where the somatosensory side is in the parietal lobe. I'll get into that in a bit. So what does primary motor cortex in that central sulcus do? Well, you get the strongest activation in an fMRI experiment in M1 if you ask people, and I'm not kidding, to do this. Right? So it does simple movement. M1 does the simplest of movements. It just has a map of every muscle, and whenever you move, there's an activation that really is like the building blocks for a more complex movement. And as movement becomes more complex, of course, you need to plan that more and more complex movement, and you move to premotor cortex, where movement plans are a bit more complex than just, right? And so the more complex a movement is, and the more it extends into the future, the stronger it is represented in terms of anterior location. Yeah? So if I decided to just step down, right, just the landing of my toes, that was motor cortex. But just the planning of the leg movement towards the floor, that sort of thing, is more premotor cortex, right? And you can imagine that just this whole idea of how am I going to walk about, that sort of thing. If I'm thinking about maybe I should walk over to the door and grasp the handle, right? That sort of thing becomes progressively more complex and more future-based. Yeah. And so planning is really the purview of the frontal cortex. And in general, there is this gradient towards more abstract and more higher order control. So very complicated decisions of, should you be writing in for brain imaging this semester, right? Very complicated, life determining choices. Those get made early in the frontal part of the brain, where you have to integrate a lot of different types of information in order to construct a valid plan. Right? So the representations in the very anterior reaches of the frontal cortex are more abstract, if you will, more personal in some way. So there's another sort of separation of the frontal lobe that I just want to start out with here. Is that if you look at the lateral side, it's usually externally driven, where there's also a medial frontal side, and that is more internally driven. So we call that, we call that hot and cold. So cold is towards the outside, it's unemotional, it's lateral. Hot is emotional. Like, how am I feeling? How will I feel if I choose brain imaging this semester? Will I feel like shit? Then I shouldn't do it, right? So it's that sort of thing. That's hot means emotional in this sense. Yeah, I'll go on, but yeah.
When you said that the frontal lobe is for highly complex and abstract processing, does that automatically imply that it's also then has a lot of interaction with, let's say, the emotional part of the brain?
Exactly. Yes, I'll get into that in a bit. There are very, very strong interactions between specifically this part, the medial part of the frontal cortex, and these motivational regions in the basal ganglia and in the brainstem. So we have really this motivational axis running into the front of the brain, from the brainstem up into the frontal lobe. Yeah. So that's what we think happens in the frontal lobe. So just reaching, grasping, that sort of thing, this emotion. And as you then, so that's when you go to the front, and when you go to the back, that's where the parietal lobe is, and that's more the sensory processing side of things. So, of course, when I'm going to move, right, I need a movement plan, but whenever I move, I will feel my finger bending, for example, right? That's a very simple sensation. But I can also, for example, when you're dancing or something, that is going to be much more complex in terms of the sensory consequences that will produce, right? And your brain needs to know what to expect in the future. So there's a very, very strong component of this type of somatosensory processing in the brain too, which exists mostly in the parietal lobe. Now you can also imagine that these two need to talk to one another, right? It's simple prediction of how something will feel, the sensory consequences of something, and the motor plan, they need to touch one another really, really intimately, right? Because they will touch one another in the outside world in terms of your interaction with the outside world. Really the interface between your plans and the feedback from your plans, from your execution of those plans, right? Sensation and action are really on the same, really organized very similarly on both sides of the central sulcus. It's a beautiful place, really. Sort of the middle of the brain, right? And it's also where we interact with the world. Now, how does this work? Like, that's just one global sort of segmentation in terms of action and perception, perhaps. And of course, this is all extremely simplified, right? There's much more to this, but general thing. If you zoom into that just a bit, and I'll see that, oh no, this is anterior, this is good, same color. So front is red and back is blue. Black is somatosensation, red is motor, right? So they are very, very similarly organized. And that's because, of course, this closeness that they need to have with one another. Yeah. What you see is that we have a full map of the body in the posterior part of the central sulcus. And we have an analog map, also a body map, we call a homunculus, on the frontal side. And that's reasonable, right? If you are going to talk and move your lips and tongue while talking, it makes sense to, at the same time, already be able to incorporate the sensory effects of that. And if you have them both on the opposite banks of the central sulcus, it's very easy to communicate, right? The axons between neurons on both sides of the central sulcus need to be very short, means fast transmission and efficient transmission, right? So you see here that the brain is organized according to what you might call its ecologic repertoire of behaviors. That was maybe a bit philosophical, but that's really in the title of papers that are about this sort of thing. So that's frontal lobe, parietal lobe, action versus sensation, right? Action on the front and sensation in the back. In general, they're not left-right, so this is just front-back. These are contralateral, as most sensation in the brain is contralateral. That is, when I move my left finger, you will see an activation in the right side of my brain and vice versa. The only very exotic thing is that the cerebellum, that beautiful region at the bottom, it's what we call ipsilaterally organized. So it is just the whole brain mirrored in the cerebellum. So if I move my left hand, we see the left cerebellum activate instead of the right motor cortex. So there's like a crossover in the brainstem that connects the cerebellum to the rest of the brain. It's insane, right? There is no logic behind that sort of thing. It's just some pre-mammal surviving and another not. Okay, sorry, I digress. So let's go into another set of really big partitions that are interesting in terms of how they map onto cognition, right? So I'm just really taking this broad view still. So what I'm showing you here is some figures from this very influential paper. It's quite recent still, by Daniel Margulies. You don't have to go look it up, but it is very influential, so I thought I'd show you. And so what they do is they say, okay, there's a, so they do a bunch of analyses, but the idea behind this is that they're identifying this region that we knew exists, a set of regions that we knew exist, which is called the default mode network. And we knew from earlier research that whenever you leave people to their own devices, which is you put them in a scanner and you just say, think about whatever, then these regions, they sort of start to co-fluctuate. Whenever you have someone perform an autobiographical memory experiment, so you ask them just to think about what their grandmother did last year or something like that, you will also see these regions, these are red here, activate. And what this paper showed, which was so tremendously influential, it's also a bit of a philosophical paper, really, is that they identified that these regions are very, very far removed from the calcarine sulcus and the occipital lobe, very far removed from the superior temporal lobe, very far removed from the somatosensory and motor cortex. And what this means is all of these regions, the somatosensory and motor cortex, the visual cortex, the auditory cortex, are where information from the outside world comes in, right? Or where we interact with the outside world. So what are these regions then? They're far removed. There are many synapses in the brain removed from the incoming information. And what that means is they represent more internally oriented information processing. Yeah. So whenever you are doing something based on memory, that is introspection, rumination, that sort of thing, is what activates these regions. So you think of these as the inside of the brain, where your I don't know what I'm saying here, but like where your sort of inner mind resides. It's a very pretentious way of saying this. It's not correct, but you get the point, right? You get sensory activations on one end of the spectrum, and you get very sort of personal internal activations on the other side of the spectrum. And you will see a lot of references. If you just go through the literature on fMRI, you will find this default mode network popping up wherever you go. It's very interesting, right? So that's what we're seeing here, right? So we have this gradient of, so you see V1 and A1 are both on one side of this first gradient, and then you have these regions, the red regions that are part of this default mode. They're far further removed from the sensory periphery, if you will. And they care about things like memory. It's very interesting. We have no idea what these regions do. Right, so for a sensation-related region in the brain, you can just show a visual stimulus and say, where do I see this visual stimulus end up, right? It's very simple. You have a lot of control, but we have very little control in terms of experimentation, in terms of what these regions do. They're Okay, yeah, it's a very sort of personal thing, right? We don't know whether it's very different, the organization in these regions, for different individuals, for example. And the locations of these regions also vary a lot from individual to individual. So these are really sort of dark spots on the map that we have for understanding the brain. Yeah, it's interesting. Yeah.
That the location varies from individual to individual. Do we know what effect that might have on the person? Anything?
Yeah, so that's people are researching this. The jury is still out. It's very open. Yeah. So no. Yeah, I could look up some literature on it. There are, so people have mapped out these regions in like huge cohorts, and then there are specific things that happen. So I think you can sort of look at the strength of the connectivity between these regions, and it does relate to things like psychotic symptoms. It does relate to things like Alzheimer's, right? So it is a very core thing, specifically on the locations and how that relates to cognition. I don't have any clear knowledge. So if you give someone LSD in the scanner, the whole sort of, so normally these regions, they all co-fluctuate, right? So something here goes up, something here goes up, something at the same time there goes up. If you give someone LSD, it sort of disappears, right? So it's also a sort of coherent syncing mechanism, it seems. But we have no idea, like mechanically, how this set of regions performs this function. So it's a beautiful, yeah, thing. It's very interesting. No idea. So we were talking about this just now, so I can be quick about it, right? In terms of motivation, right, our propensity and capacity to act in the world, that of course depends on how you feel about what results your actions might have, right? Should I punch this person in the face? Mostly no, right? Because there will be consequences, right? And you need to intuit those consequences in an emotional way, right? Because you probably get beat up yourself, and that is going to be painful, and you can, intuit that will not be able to plan, right? So you see how, it's a very bad example, of course, but you see how your emotional state interacts with how you are planning your future actions in this way. And there is a specific brain axis that gives rise to this, right? So you have this set of regions, again, in that set of regions called the basal ganglia in the middle of the brain, that interact very strongly with the frontal cortex. And this circuit is a whole, so that is the frontal cortex that thinks about the more abstract things, and this sort of intuition, sort of self-empathy, would my body feel about it, sort of thing. That all needs to be integrated, and it is in this loop. Now, there are a few neurotransmitters that are specifically important in this type of thing, things like action planning, and they are dopamine and noradrenaline. I'll go into those in a bit later on. Forgetting anything? Yeah. So just think about this as, should I stay or should I go? Right? Whenever you decide to do something, it's generally that sort of thing is computed in this circuit. Whenever you decide to not do something, it's generally also computed in this circuit. It's just that, the balance just falls out a bit different. Now, just one step back to microscopy, right? We have these neurotransmitters that I just named, right? So dopamine and noradrenaline, a bunch more, right? Serotonin, acetylcholine. I used to mostly know these because there's drugs that mimic the effects of these neurotransmitters. And so those are a specific type of neurotransmitters. They are metabotropic. I'll go into what that means. But the most pervasive neurotransmitter, trouble finding words. So most of the neurotransmitters in the brain, they are not what we call metabotropic, they're called ionotropic. What that means is that whenever they are put out into a synapse by a cell, that neurotransmitter is picked up by what we call the postsynaptic cell, so that's the receiving end cell. And this this membrane protein is sensitive to the appearance of this neurotransmitter, and it will open up the membrane. So we will have a direct electrical effect. It will change the electrical properties of the receiving cell directly by opening up its membrane and letting ions flow through the membrane, which changes the electrical potential of the membrane. So if you don't know what an electrical potential is, don't worry about it. Ionotropic receptors, such as those sensitive to glutamate and GABA, directly impact whether a cell will fire or not. So if glutamate reaches a postsynaptic cell, it is more likely to fire. And if GABA reaches a postsynaptic cell, it is less likely to fire in a very, very direct way. So this is something that happens in milliseconds. Very directly, if a volley of glutamate arrives at a postsynaptic cell and it's timed just right so that all of these synapses get activated at the same time, that is the specific type of moment where that postsynaptic cell will fire an action potential of itself and thereby propagate the signal further into the brain. So that's how that type of neurotransmitter works, nice and fast, and everything you see is the result, like everything you see right now, all of these photons hitting your retina, and then you seeing them, that is fast and the result of these types of transmitters. But, of course, some of you may be turning a bit drowsy now. Yeah. Now, that type of slow fluctuation is more metabolic, if you will. Yeah. And that type of change, things like emotional state, things like arousal, that type of, whether you're paying attention or not, that type of thing is organized through these metabotropic receptors, which stand for, like, think of it as metabolics, right? Metabolism. They change your homeostasis. Again, a difficult word, sorry. What they do is they act slowly and they just change the settings of the system. You, for example, be able to pay more attention, or to be able to run faster, right? Or be happy, or, you know, whatever. So these very slow types of changes are mediated through these metabotropic receptors, and examples are dopamine, noradrenaline, serotonin. So what these do is they, when a neurotransmitter binds there, they don't open a channel directly, but there is a whole complicated transmission chain that changes. You might even get changes in how genes are being expressed, all on slower timescales than the direct transmission of electrical signals. Clear?
Do these two compete with each other?
Not directly, I don't think. But there is a bunch of overlap. For example, there are GABA receptors that are direct ionotropic receptors. There are also GABA receptors that are metabotropic. So just separating things out is always a caricature, and you caught me. Yeah. So there are these sort of dichotomies, these discrete things that I'm saying. They hold true at some level of detail, but they start to break apart when you dive in deeper. So these don't really compete because they, in essence, exist on separate timescales.
So one, the GABA thing is more instinctive or spontaneous, whereas the other one is more over time.
Yeah, slower in terms of function. Yeah, and you can imagine that it makes sense to have fast depression of signals versus slow depression of signals. This is a GABA is a negative, right? It decreases the chances that a neuron will fire. Yeah. But this means that these are very important, but both in different ways, right? So if you block a glutamate receptor, you have problems because signal transmission, direct signal transmission, starts to cease, right? That can be problematic. So it could be toxic, for example. Yeah. This is much slower. So things like antidepressant medication, for example, one of the major classes is selective serotonin reuptake inhibitors. Well, serotonin is in there. It does something with the reuptake of the serotonin from the synapse. You get the point, right? Different timescales. Let's not belabor the point too much. What is interesting is what these metabotropic receptors do. So they fine-tune slowly our behavior. By slowly, I mean anything from like around a couple hundred milliseconds to longer. Yeah. because the time scale at which the inotropic receptors work is really like millisecond time scale, tens of milliseconds perhaps. What's nice to see is that these different neuromodulators, as we call them, the metabotropic ones, they are organized quite differently across the cerebral cortex and the brain in general. So here's an example of dopamine. It's generally released from this set of regions called the ventral tegmental area and the substantia nigra. They're in the brainstem. You can actually see them. Substantia nigra means black matter. So you can actually just see it if you cut someone's head open, you can see a little black thing there. That's the substantia nigra. That starts dying off in Parkinson's disease, for example. VTA. So this is like a motor region. That's why Parkinson's patients have motor problems. VTA also projects dopamine, but to slightly different parts of the brain. And those are more like the addictive substances substrate of the brain. So like the pleasure center and that sort of thing. So these signals, they end up going from this brainstem region to the basal ganglia, and then up into the frontal cortex and then throughout the brain. So this is really that motivational axis of the brain, like so. That's really strongly dopamine based. Now you might think, okay, that's all nice and good, but we're interested in this fMRI thing, remember? And that is, we want to see a relation to direct neural firing. So maybe we're not all interested in whatever dopamine might do. It turns out that we can't really say that sort of thing. People have done experiments where they inject dopamine into dopamine agonists into macaques in this case, and what it does is it has a profound influence on whether the bulge response that you measure reflects underlying neural firing, right? So this indirect thing that we've already identified, we're looking at blood, we are not looking at neurons firing, right? There isn't even a fixed relation between these two, even though we might like to assume it, and it is modulated by dopamine, by these types of neurotransmitters. So yet another layer of uncertainty in the way that we are using the fMRI scanner, and it's because of the function of this type of neurotransmitter. So this is really reward related and movement initiation related dopamine. Separate system that I think is quite interesting in this context is noradrenaline, which is very arousal based. So some of you I can see here have low noradrenaline levels. Now, noradrenaline levels are mostly coming from this tiny thing here in the back of the brainstem. If you want to do fMRI on this LC region, which is called the locus ceruleus, which stands for blue spot. So that when you cut people up with a certain staining, it's blue instead of black. And that has these really, really long axons that project throughout the brain. And so whenever you're about to wake up, that is mediated by a boost of noradrenaline from the locus ceruleus to the rest of the brain that increases arousal in all brain areas. And what I mean by arousal is that, for example, your auditory cortex will start picking up sounds much more sensitively and thereby waking you up from sounds that start occurring, right? So it's related to things like circadian rhythm and that sort of thing. Yeah. You see, it goes to these low-lying regions here. So if you're more aroused, you might remember things better. That's because the LC projects pretty directly to things like the hippocampus, memory-related regions. But of course, if you're highly aroused, that will also change your planning, for example, your estimates of how far you could run, that sort of thing. But it also changes your perception even. So there's studies showing that high levels of LC activation really change how you make sensory decisions. So what we think is that noradrenaline really changes a very basic setting of how the brain processes it. Sorry, go ahead.
Do you have a sort of say temporal tolerance to adrenaline? Like, do we keep being aroused for a longer time? Would you need more of it, or do you just need it?
Yeah, in general, that is how many sort of neurotransmitters work. Whenever you have a large inflow for a prolonged period of time, that will sort of overstimulate, of course, certain brain regions, certain postsynaptic cells. And what one typical response that these cells would have is that they would downregulate the amount of receptors that they express in their synapses. So they would sort of make themselves less sensitive on the long term for the impact of that neurotransmitter. But this is like a general thing that happens. Yeah.
You spoke about the LC and how it is connected to waking up with increasing the voices and stuff.
Yeah.
When people have difficulty sleeping, would suppressing the LC with, say, the reduction of noradrenaline also cause them to sleep better, sleep longer?
I don't know if it would have that direct clinical impact, but I'm pretty sure, and I don't think this experiment has been done, but I'm pretty sure that if you would inject like inhibiting substance, right? So that if you could silence the LC, you could do that and people would just fall asleep. Or at least tumble over, or, you know, it would be very drastic. Yeah. Yeah, definitely. But very coarsely. Yeah. I just drank coffee, right? What that does is it also has similar effects to noradrenaline throughout the brain, right? So that's why people use it to concentrate, increase their arousal. These two these two neurotransmitters are very strongly activated by things like amphetamines, that sort of thing. So there's very, yeah, really, you know, regulating up your levels of arousal and activation, your response drivenness, or your reward drivenness, if you will, or yeah, down regulating them. Yeah. I'm at my last slide, so we're good on the lunch side of things, I think, which is always very rewarding. Any questions? Yeah. Yes. So yeah, that's a good question. So inside the here in the basal ganglia, there is this circuit, right? And I didn't really get into it, but you saw that there were like a lot of different regions there, right? And so one of the there are a couple of pathways in the basal ganglia. Some are they're like a go pathway, they're like a no-go pathway, and they sort of keep each other in balance. So whenever you decide to do something, the balance just shifts, and it's very easy for the entire system, if you will, to then partake in the action. And so this balance is a very sort of natural, very delicate thing, and it's partly mediated by dopaminergic transmission. And so what you have is that one side of that equation, if you will, is altered. It doesn't receive as much input, and then the other one can sort of break through, right? And that means that then another feedback loop gets activated, which tries to still down the movement again. And so in general, what you have is that the initiation of movement is impeded in Parkinson's because of this lack of dopamine. And at the same time, this whole dysregulation of this delicate balance causes these tremors, because just maintaining a still posture is actually not a passive thing, it's an active thing. And so the dysregulation of that active process causes these tremors. Yeah. So it's just this different feedback loops. It's also why I keep talking. I'm sorry for all of you that are hungry. It's also why if you do deep brain stimulation works so well in Parkinson's disease, if all medication is exhausted. So you stimulate a specific motor nucleus inside the subthalamic nucleus. And if you do that in the right way, it will actually sort of reinstate this balance, sort of push back the thing into a sort of non-tremor state. So you really have to like sort of engineer this sort of feedback loop circuit inside those basal ganglia to, well, at least alleviate these symptoms, not fix the disease, of course. Yeah. Excellent. It's time for weekend, lunch and weekend. Yay. Just keep letting me know if anything is unclear in the practicals, in the quizzes, right? I just type stuff, right? So sometimes I say things that aren't so clear, for example. Yeah.
Yeah.